Hi, everybody. Could you hear me?
Yes.
Okay, start to my presentation. Hi, good afternoon, everyone. Welcome to my PhD defense. And the title of my thesis is cytoskeletal proteins, MREB, and FTSA and the peptide glycan hydrolysis, PPP4, act together in longitudinally dividing bacterium candidates, psilocybin honesty. And in this presentation, I will introduce the background and make a brief review of the thesis. While the bacteria are one of the oldest unicellular life forms, their relatively simple structure provides a model system to help us easily understand how cells work. Cell division is one of the most important progresses in their bacterial life cycle and was studied for a long time. This drawing sheet shows the division process of a typical rod-shaped bacterium, which is better studied compared to other bacteria with different shapes. Normally, a rod-shaped bacteria, it will grow in length and divided in the middle. During the cell growth, the cell will become longer and the chromosome replication will be accomplished. The cytokinesis, the cleavage of the cytoplasma, is initiated by example of the ring, which is a structure in the middle determining the division site and serving as a scaffold for division machinery. The ring then constricted to bring about their division, and the cell wall synthesis follows the ring in length. The investigations for the model bacteria, it contributes a lot for our understanding of bacterial division, but the non-model bacterium, they are also very important for us to review possible alternative mechanisms and expanding new bacterial applications. And my thesis is mainly focused on a non-model bacterium, the Candidatus psilocybin honesty. And this bacterium, it is an unculturable rod-shaped gamma proteobacterium. It is the ectosymbiont of the nematode host, Laxis odysseus, like this image shows. This bacteria lives on the cuticle of host and forms a single layer coat perpendicularly to the surface, which you can see from this image. And this symbiotic bacterium is divided by a longitudinal fissions, which means their cells grow in waves instead of elongating and constrict along its long axis during the cell division. Its special mode offers us a unique opportunity to determine their flexibility of division mechanism and to gain a better knowledge about the fundamentals of their fission process. So to understand whether the longitudinal division mode is achieved by reconstruction of a known system or new mechanisms, two cytoskeleton proteins and a cell wall hydrolysis was investigated in my thesis. And these three different proteins, they are the key factors of ring example cell growth and cell wall synthesis, respectively. For the experimental chapter 1, it is about the tubulin homolog protein FTSA. FTSA can polymerize into filaments to form the Z-ring structure, and the FTSA of T honesty was characterized in vitro and also in E. coli cells. We discovered, based on these different experiments, we discovered that the FTSA of T honesty is naturally sensitive. It's naturally sensitive to temperature, which might be adaptation to the growth temperature of its host. And the second experimental chapter is about the acting homolog protein mREB. The mREB of the honesty, it was localized in fixed cells, and it shows actually a 90-degree rotate with respect to other root-shaped bacteria like this. At the division pal, the mREB also co-localized with FTSA and in the honesty, and also accumulates around the future septum size preceding the localization of FTSA, suggesting it possibly play a role in division pal determination. And for the last experimental chapter, it focused on PPP4. It was PPP4 was immunolabeled in cells and showed the polar localizations. You can see here, and also accumulates at the constriction site like this. And the isolated PPP4 proteins can form a dimer in vitro, and also it shows different selectivity for beta-lactamase compared to its homolog from E. coli. And at the end, the localization model of these three proteins was concluded, and we also compared this model to the growth model of cocci as Formonian. Yeah, that's all about my thesis. Thank you for watching.
Thank you, Dean Wang. We will now wait until 3:15 when the defense begins. Meanwhile, I would like to ask all committee members and yourself to keep your camera and microphone switched off. And we will continue at 3:15. Thank you. So if the screen can be removed, it's 3:15 and we continue with the defense. And I would like to ask all committee members to switch on their screen. And I happily give the word to the rector.
Ready for it? Okay. Ladies and gentlemen, welcome to the special meeting of the Doctorate Board of the University of Amsterdam on the occasion of the PhD defense of Yinlang Wang. Dear candidates, to open the discussion, please read aloud the first part of the formula.
By authority of the rector, magnificence, and the doctorate board of this university, I shall, during this hour, in order to obtain the doctor, publicly defense my thesis entitled The Cytoskeletal Proteins MREB and FTSA and the Peptidoglycan Hydrolysis PPP4 act together in longitudinally dividing bacterium candidates through symbiont honesty. I entreat all these who wish to voice criticism of the contents of their thesis to do so in the truthful and lucid manner, so as to provide an opportunity for an ordered exchange of views.
Thank you. I hereby introduce to you the first opponent, Professor Dirk-Jan Scheffers, Professor in Molecular Microbiology from the University of Groningen. Professor Scheffers, the floor is yours.
Thank you, Mr. Rector. According to my right, but not without permission of the rector, I'd like to discuss your thesis with you, dear candidate. But first, I would like to compliment you on completing this thesis. It is fascinating work about very special bacteria that only grow on nematodes and that divide in ways that for a long time we thought not possible, because we always thought that bacteria would go for the shortest route to divide. And these bacteria do exactly the opposite. And you cannot grow them in the laboratory, which makes it even more difficult to do these studies. So I have taken my hat off, but hats off to you for producing this work.
Thank you.
So I would like to go with you to chapter 4 and discuss a number of things about chapter 4. So in the beginning of chapter 4, on page 92 and 93, you describe the cell wall of the candidatus Theosymbion onesti. And in table 1, you list the composition of peptidoglycan in terms of monomers, dimers, and multimers. My first question to you is, this is a gram-negative bacterium. What can you tell me about the composition of the stem peptide of this peptidoglycan?
Stem peptides. For the stem peptides, do you mean the component of peptides?
Is it, yes, does it have like mesodiaminopimelic acid at position 3, or is it different from standard gram-negative stem peptides?
Well, it's a gram-negative bacteria. I did not see special peptides in in our results, we only have this one analysis, so I assume it should be close to standard peptides.
Okay, but you cannot make a distinction because this was mass spectrometry, the analysis, correct? Okay, so you cannot, then it might be difficult to assign all the masses specifically. I was wondering if it's possible. Okay, then I want to go with you to figure 3 and the top left corner.
Yeah, so figure 3, top left corner.
So you see the PBP4 immunolabeling.
Yeah.
And you see 2 spots.
Yeah.
Where would you say these spots are?
Where?
Yes.
When you mean where, you mean from the top well or from the front well or distribution? Sorry, I do not understand.
Sorry, how would you call this localization? I think it is at the pole, right?
Yeah.
Yes. Okay. So
you want me to describe if this is poor or right?
Well, what I would like to ask you is a bit of a philosophical question. What is a pole in your view?
Oh, that's really a question. Well, I understand because even from the beginning, we can just think from another side. We do not treat it as a pore because normally we think narrow area is pore, but it's not really true something because the pore there is not really a definition, and if we just think it's a narrow area, it means nothing because it's different from species to species. In this case, and from the aspects of the division, I think you can think it's not real a pore in this case, and also we do not have any evidence to prove this is the real pore. We do not have in some bacteria, the pore, it has special structure and special localization of some proteins, and all of this, they did not exist in Candidatus T. honesty. So for division and in this case, I think we can say it's not real pore here.
Yeah, it's a funny way of dividing in that way. So if an E. coli cell divides, we call where it has divided the pole. So in this case, it would be actually somewhere in the middle or along the longitudinal axis of the cell. Okay, thanks for that. So now I would like to go with you to back to figure 2, where you use the antibody against PBP4 to show that the E. coli antibody, that you can label your protein both on a Western blot and in microscopy.
Mm-hmm.
And further in the chapter, you use fluorescent bocillin. My question is, have you ever tried to label all the penicillin binding proteins of Thiosymbion onesti with bocillin in one go so that you get a pattern?
I tried. Well, we did not successfully get the results. The reason is the amount of cells we have. Because it's not cultural, so we cannot have a cells we want. The material is very, very limited. And for bacillin, we first tested in E. coli, and the amount of the protein to show a bacillin signal in my lab is not so bacillin is not very sensitive in our lab, and we need a lot amount of the total proteins to get all signal bands, and we cannot get enough material for honesty cells. So I just use a little bit to try after some other experiments. So I just reuse the material to have a try. I just get nothing. So we don't know like how many real PPP proteins in their T-honest candidates T-honesty because all now all the information comes from their analysis, so it's not true. So I have to say sorry, we don't have results for that.
Well, your explanation is very clear. So I want to thank you for your answers, and I give the word back to the rector.
Thank you very much. With that, I want to give the floor to the second opponent, Professor Silvia Bulgaresi from the University of Vienna, professor in environmental cell biology. Professor Bulgaresi, the floor is yours.
Thank you. It was a pleasure to read your thesis, Jinglan, and congratulations again for daring to work on this very difficult system. So I join Professor Scheffer in his praise. So my first question is, why do you think these bacteria evolve longitudinal division?
Oh, sorry, could you repeat?
Yeah, could you speculate why these bacteria evolve this extraordinary way to divide? Why longitudinal division evolved?
Oh,
maybe you can think about if you know other organisms that divide longitudinally, and maybe by thinking about other organisms that divide longitudinally, maybe you can speculate it about what could be the advantage of such division.
Division.
Do you know other bacteria that divide like this, or other even eukaryotic cells? Or
well, of course, we have another one very close to this, the Promotieceremonie, but they are too close to each other. So I think we can put it into the same type for both of these. But for other species, now the information for honesty, I think it's still very limited.
Okay, I can help you maybe. So there is another bacterium which is attached to a ciliate, also in the sea, and is also dividing longitudinally. And then there are also have been recently described that like bacteria that are attached to the cavity or an oral cavity of warm-blooded vertebrates. They are also dividing longitudinally. And also, so what do they have in common, all these bacteria?
Oh, you mean, I guess you mean because they are all symbiont for this kind of situation? they can easily attach on their host. I guess that's the answer you want to know.
It could be, right? Because of course there are also bacteria which are attached to hosts and are not divided longitudinally. But up to now, there is this common theme, if you want, of longitudinal division and symbiotic lifestyle. So
yeah, that's
right?
really, yeah, of course that's possible.
Yeah.
I actually always thinking if they want, if the bacteria they just want to keep attached on their host, well, compared to change a division mode, they just use some other ways to attach. Is I think it's easier for them, and also so I don't know if it's a real reason for this special division, but of course it can be explanation for this division.
Yeah, it's pure speculation, but it could be. And if we assume that longitudinal division evolved from transverse division, how could this have happened? So how could you imagine that this type of division evolved? Neke wrote a beautiful review article about two possible scenarios in 2013. Maybe you remember, about how two possible ways we can envision.
Sorry, could you explain your questions a little bit more? I'm sorry.
Yes. So there are two hypothetical scenarios, okay? How to explain. So I tell you one of the two, and maybe you can think and the other. So one could think that the all cytokinetic machinery turn 90 degrees, right? Mm-hmm. Or what could we think? What would be the alternative scenario?
Well, now I think it's not a real shift. So oh, you mean they just like reshape their pipette like,
right?
They become narrow and become very long.
Yeah.
So, and also that's why they started to longitudinal divided, and so they just change their lines and into a very short distance. Wow.
Yeah. Okay. Your answer. Okay. And maybe another question. So,
please, a short one.
Yeah. Are there bacteria? So is the cell wall essential? Are there bacteria? Can bacteria exist without a cell wall?
Well, normally no. And also in this case, of course, no. And for bacteria, only few of them doesn't have their cell walls. They're always in very special conditions. And if they don't have cell walls, they cannot resistance to their omestic strains, and also they will, and also they cannot live well in the environment. And for honesty, it lives in the oceans, and which I think their cell wall is very essential for them.
Okay, thank you.
Thank you, Professor Bulgaresi. With this, we move on to the third opponent, Professor Gerard Muizer from the University of Amsterdam, professor in microbial systems ecology.
Thank you very much, meneer de rector. So Dear candidate, I want to compliment you with your thesis, which I've read with great interest. But I also have some questions and I want to have a discussion with you. How does the nematode get its symbionts?
How does this nematode in symbionts?
Gets its symbionts.
And how does while the bacteria, they lived on their skin and they live together and they share their I think there is material exchange between them, and also they benefits to each other.
But I mean, on page 10, you mentioned the secretion of a calcium-dependent monospecific lectin.
Yeah.
Mediate association.
Or you mean the way they
Yeah, the way, how do they get them? Because they are probably, let's say, naked for some time, and then they actually can bind them. And I assume when they have this lectin, they can get them from the environment.
They actually, I think they can get it from the environment. So if we separate the bacterium and the worm, and if they have the lectin and this receptors, they will attach to each other and start to grow together. So that's possible to.
So they are free living bacteria in the environment. Yes.
They're not free living, they'll always live together, but in laboratory conditions, if we separate, they can get it freely. So, and also the newborn of the nematode, they don't have the symbiont. So, the way of a newborn nematode get it, we guess it should be from the environment, but later they will live together forever.
Yeah, but do you know if scientists have detected them in the environment?
Detective the nematode or the...
No, not the nematodes, the free living symbionts.
I don't think we get free living bacteria in environments.
Okay.
No.
Okay. Okay, I will go to the next question, and I might come back to that. I mean, how would you name the interaction between the nematode and the bacterium? How would you name this?
Name the interaction between the nematode and environment.
Is it a symbiosis or a mutualism or a syntrophy?
How we name the interaction?
So what does the bacterium give to the nematode?
It's still unknown. I think it's still unknown. How does it give to?
Oh yeah, I mean, it can fix CO2 and it can fix nitrogen. So I assume that it gives carbon and nitrogen to the nematode, or not?
It is believed that it can give some.
Okay, but then my question is, what does the bacterium get in return from the nematode? Because if you would call it a symbiosis or a mutualism,
Oh, okay.
Then, of course, there is a kind of win-win
And
situation.
Yeah, okay, that's why we name it similar, because it benefits to each other in some way. Well, we don't have a real evidence about like how what the bacteria can get from the worm. We assume the worm like can help the bacterium to move into the environment with more sulfur, and it can cross the layers with different concentrations of oxygens, which will help the oxidizing of the sulfur for bacterium to get energies, and this energy more probably returned back to the worms, and that's
I think so too. So by moving up and down by the nematode, it might come in a sulfidic zone where it takes up sulfur or sulfide, and then to the yoxic zone where it can oxidize sulfide to elemental sulfur. Yeah. Okay. Yeah. So I have another question. So the candidatus thiosymbion is an ectosymbiont for the nematodes, but also an endosymbiont for the annelids. Yeah. Are the endosymbionts all of the annelids also dividing longitudinal?
No, they do not divide longitudinal. I think, yeah, they don't. For the endosymbionts, they divide it normally.
They don't. Okay, they really divide. Okay, good. So, why do some nematodes contain endo and other, let's say, annelids, other ectosymbionts? What would be the advantage of an endo or an ectosymbiont?
Advantages of endosymbionts. I have to say I do not know much information about the endosymbionts. I don't know what they can get from the host. What the host can get from them.
Probably the same. They might also get, let's say, carbon and nitrogen.
But if that is the case, the endo and extrosymbionts, the function of them are reductant to each other. And also for endosymbionts, it's not easy to get a sulfur from the environment. So I don't.
I don't think so, because I mean there are more, let's say, worms or bivalves which are endosymbionts and get the sulfide. The sulfide actually just diffuses through the membrane, and so therefore also the endosymbionts will get their sulfide. Yeah, so I think that the function, as you mentioned, is indeed the same. But why, let's say, some of the same, let's say, genus, Candidatus thiosymbion, would become endosymbionts or ectosymbionts? That is a kind of a question. Yeah, Meneer de Rector, can I still have one question or not? You're mute.
Could you hear me?
Yes.
Well,
There's please a short question and a short.
Okay. So you have used E. coli as an experimental host, yes. But is that the most, let's say, the most ideal host? Do you think that it's the most ideal host?
Well, it's most common use. So
yes. It's the most common use, but that does not mean that it's always the most ideal, because
probably most not ideal, but we don't have option in our case. So
I mean, would it be possible, for instance, to use an artificial cell to study?
Okay. I think well, the E. coli here, it's not really a host. It's just it's just something try to so it's a tool. It's a tool to express the the proteins is not a real host, so it not
Okay.
work as a host.
I can mention to you that 2 days ago there was a paper in Nature Communications in which they used lipid vesicles with the five genes, and they actually saw that it worked, and so it might also work for your organism. Yeah. Okay, meneer Director, thank you very much.
Thank you very much for this last suggestion. With that, we move on to our next opponent, which is Professor Stanley Bro from the University of Amsterdam, professor in, I should say it correct, molecular biology and food safety.
Thank you, Mr. Rector. According to my rights, but not without permission of the rector, I would like to discuss with you, dear candidate, on the content of your thesis. Very nice work, quite challenging work also, and novel work, which is really good things for a thesis to have, of course. And I usually start at the end. And page 120, you have a scheme, a model that you propose. Can you talk me through this model in terms of, where is the same as Streptococcus pneumoniae, because that's the comparison, and where will it differ, what you have seen? Where might it differ from this Strep pneumoniae?
Okay. The reason we compared it to Streptococcus pneumoniae actually is related to the question Sylvia mentioned before, because we don't have a better explanations now, so we just assume this model that's the spectrum become extra long and the cells just narrow their cell lines.
Right.
So from the top wheel, it's a round shape, so it's a coccus from only the top wheel, and that's why we compared to both of them. And so if we just from the top wheel, we can think the pneumonia and the symbiont, they actually have a very, very similar model.
The shape.
Yeah, the
in
shape.
terms of the shape it's the same. Yes.
The lateral wheel, the only difference is the size of the spectrum, yeah, that's her only difference. Probably it's very challenging to finish this big septum, but it can do it.
So you start by observing very well into your cells, and then you compare the shapes of the cells and see, well, there are similarities and differences. I see indeed you have indicated the two proteins FTSZ and MREB with these nice colored lines. How, because I'm a molecular biologist or biochemist, so I'd like to come to the components in Candidatus Tionesti itself. So how would you do that? How would you, because you have studied two components, MREB, FTSZ, but that would not be the only thing that is present in the organism. Could you think of a way in which we can get the interacting partners in the organism itself?
Partners. Well, I think it's so. I think we can use cross linkage. So some chemicals can make the proteins cross linked to each other, and we actually could have some live bacteria and worms. And I think that if we cross link the different proteins, well, like for example, FTSA, I think we use some chemicals to cross-link the proteins, and we can use antibodies attached on the column and use a special column to collect the FTSA and the proteins linked with FTSA will all live together, and we can send them to do a mass spec, and we will.
I like that idea. That's a nice idea. These are good ideas indeed. And in terms of antibodies available, I mean, I am not from this field at all, So I was just wondering, do you have, or are there groups where there are antibodies against the native proteins that are sufficiently cross-reactive?
Because their FTC and the MRB, they are conserved to each other. So what the antibody we use is actually their polyclonal E. coli antibody. But it is possible to select the antibody which only interact with symbiotic FTSA. There is some different way to purify their antibodies, and we do have enough antibodies, because for E. coli and the MRB and the FTSA antibodies.
Right.
Yeah.
Would it be possible, if I may interrupt you there, would it be possible to go and also select the different stages, because you indicate different stages on the cell cycle here? Yeah, I mean, the non-divided cell, the cell on the way to division, and the divided cell. Would it be possible to select these stages in your views in some way, and then actually do these experiments on these different stages?
It could be difficult. I think the first thing is that for for a lot of model bacterium, when we do it in lab, there are always a lot of dividing cells and also non-dividing cells. We have a different cells in different stage is a lot, and for honesty, we don't have enough measure. And the second thing is that actually the dividing cells in honesty is only it's less than 10%, is very
exactly
low. So it's not active dividing cells. So probably.
Thank you very much. Thank you very much. It was a very good answer. It's a low amount of cells, the actual dividing ones likely. But I was just wondering whether you had considered at some point to use flow cytometry to.
Cytometry.
Yeah.
Third question, please.
So whether flow cytometry would be possible to isolate.
Flow cytometry could separate every kind of cells in theory, and of course in this case. Probably we can try in the future. Yeah.
Thank you very much. I hear the rector somehow. So is that correct?
Yes, that is correct.
Well, I give the word.
That was a nice short question and a nice short answer. Thank you very much.
Okay.
That's going to leave the floor for the last opponent.
Yes.
Dr. Maaike Stam, Associate Professor at the University of Amsterdam, working on gene regulation and epigenetics. Dr. Stam, the floor is yours.
Thank you. Dear candidates, I also want to congratulate you with this very interesting work on a very challenging subject. I have a few questions to you, and the first has to do with FTC, has to do with chapter 2 and chapter 3. Because in chapter 2, you mentioned that FTSZ from Honesty can assemble rings in E. coli, but they do not localize along the longitudinal axis like in Candidatus Honesty. And then you also say that the localization is likely to be dependent on other regulatory proteins. Then in chapter 3, you show that MREB from Honesty could actually be this protein. What do you expect to happen if you would actually use E. coli and then swap the protein MREB from E. coli with MREB from Candidatus Honesty? Would you think that is sufficient for longitudinal division, or do you think there's other proteins that needs to be transformed as well from Honesty?
I think MRB is not enough for this. It should be need others. because we can see that the localization of MREB it's not as dense as the FTSA in their constriction area. So they do have their interaction, but in E. coli, the interaction of FTSA and MREB is not strongly and only happened in special stages. And in honesty case, I think it just helped their division site determination, but for the real for the localization and the other regulations, I think there must be a system to do this. MRB is should not be enough for FTSA to localize properly in all.
So what? Yeah. So what type of experiments would you propose to perform to find out which proteins are needed in addition to MREB? And do you think E. coli would be the model to use, or would it be maybe the model that was indicated by Professor, what's it now, Muizer? He came up with with another model, but maybe first answer. So, what type of experiments would you propose to perform to find out which proteins are involved in addition to MREB?
Initiate MREB.
In addition, on top of MREB. What type of experiments do you think of to figure out which proteins are needed in addition to MREB from ONS?
Well, all the protein we know they to help FTSA to help FTSA localization, we do not find homologies in Luxis. So I think the only way to know what is FTSA need addiction is just like the question asked by this Professor Stanley. We need to find new proteins, probably unknown proteins, interact with FTSA, and that could be what's FTSA needed in addiction, except MREB. And
okay. Thank you. I have one more question, because you show that FTSC is temperature sensitive. And you say it could be an adaptation to the temperature at which the host grows. At the same time, so in line with this, is this nematode to which it attaches, is that an obligate symbiont? Does it need this bacteria to live? Because you also say that the nematode cannot survive above 41 degrees. And so I was actually wondering, in case it is an obligate symbiont, then it could be that the temperature sensitivity of the nematode is due to the temperature sensitivity of the bacteria. Is that a likely idea? Or does it make any sense? Or
well, I kind of I think I kind of agree with you. I think because for other nematodes, a lot of them can be resistant to the heat, and the 41 is not really. 42 is not really high degree, and they live in tropical conditions, which is always warm. So actually, I think it's a kind of make no sense that the nematode cannot survive in high temperature. So I think it makes sense for me, but I'm not a specialist for nematode. So I will I will ask some of my colleagues but in my idea, I agree with you. So I think
Hora est. Mr. Rector, you are muted.
It happens to me all the time. The discussion is closed now, and I request the candidate to read the second part of the formula.
having thus concluded the defense of my thesis to the best of my ability and pending the decision of the doctorate committee, I should like to express my thanks to my highly esteemed supervisor, Dan Kadal
Thank you.
in particularly, as well as to those who have courteously brought forward their criticisms, and to all who have honored this ceremony with their presence. Thank you.
Thank you. The committee retires for deliberations. For those online, I would say stay tuned.
If the committee is back, I will give the floor to the rector and the screen can be removed. Thank you. So the floor is to the rector again.
Thank you. Esteemed candidates.
I didn't see the candidate.
I see the candidates.
Where is the candidate?
That indeed is a good question.
Maybe we need paronyms after all.
Yes. It's of course late in China. I don't know what time it is now.
Sure to leave. There's candidate. Yeah.
Oh, okay.
It's arriving. It's arriving.
Jinglang, can you switch on your camera, please?
I cannot switch on my camera because it was stopped by the host. Could you help me to set it?
Okay, Fabian, can you switch on the camera of Jinglang?
Yeah, it works.
Okay, great. Again, the floor to the rector.
Thank you. Esteemed candidates, after due consideration of the thesis that you have submitted, we have decided to grant you the doctoral degree. The supervisor, Dr. Ten Brouwer, is authorized to confer this dignity upon you in the customary fashion.
Okay, I most willingly accept the task assigned to me by the Rector Magnificus of the University by virtue of the power conferred on us by law and in accordance with the decision of the Rector and the Doctoral Board of the University of Amsterdam. I hereby confirm upon you, Jinglang Wang, the degree of doctor, and grant you all the rights pertaining thereto by law or custom. In evidence hereof, you will be presented at a later stage with the degree certificate signed by the rector and the supervisors and confirmed with the Great Seal of the University. Having thus fulfilled the task assigned to me, I have the honor of being the first to address you as doctor and to congratulate you with the dignity conferred to you. Dear Dr. Wang, dear Jinglang, you started your PhD project in September 2015 in our group at Science Park. And while you started to get acquainted with the city, the building and the group members, the permanent employees of the group were busy preparing the move to the new built O2 building of the Free University. We moved in March 2016. And as is normal for new buildings, it took some time to get the lab up and running again. Because of the distance, you also moved to a living place more nearby the O2 building. About a year later, in October 2017, we had to move back to Science Park and install the lab again at the new location, and you also moved back to another house. Although the result of the relocations was an everlasting friendship with our colleagues of the Free University, it also made efficient experimental work challenging. Finding the right person in the right place to ask for information or help is not easy when you come from China and has to adapt to a new language, customs, and even your supervisors are new to the place and therefore struggle with what to find where. That must not have been very easy for you. However, you did your experiments as if it was business as usual. The next unforeseen development was the arrival of Corona and the lockdown. You had to work in shifts in the lab to make the 1.5 meter working distance possible. This not only made the work difficult, it also reduced social interactions among the group members. As you were writing at that time, you often had to stay home and suffer loneliness in a confined space. All in all, not strange that the whole PhD project took more time than we all wanted and that you left for China to finish the writing. Your task was to find out whether the fission of the longitudinal dividing bacterial species Candidatus theosymbion or nasty required a similar peptidoglycan machinery as E. coli that divides perpendicular to its length axis. To get you familiar with the techniques we frequently use, you started with the localization of the actin homologue MRAB in the symbionts, which Sylvia Bulgaresi from the University of Vienna was so kind to send us. The audience should realize that the bacteria cannot be grown in the lab and are collected in Belize. We tried to grow them with the help of Professor Meuser, who is also present in the committee. And although we encountered many interesting species in the sample, the symbionts refused to grow. We are also very happy that Professor Bulgaresi is present as a member of your defense committee, because together with Sylvia and especially Nika Penda, you found out that MRAB is involved in peptidoglycan synthesis of the cell division plane, which was published in Current Biology. It also took you to Vienna together with Yolanda to learn more super resolution microscopy skills. Next, you set out to make antibodies against the putative cell division protein PBP3. Unfortunately, this project had to be abandoned as the antibodies did not recognize the protein in the cells. Another part of the project was to find out whether the symbiont cell division protein FGZ deviated in properties from that of E. coli FGZ that could explain part of the difference in mode of growth and division of the symbiont. You published the results of this study in the Journal of Antibiotics. The last project you took on was the characterization and localization of the endopeptidase PP4, which you published in the International Journal of Molecular Sciences. I very much want to congratulate you with this achievement, and also I want to thank your husband and your parents for all the support you got during your stay in the Netherlands. I want to thank you for all your hard work and determination to get your PhD work finished, despite all the moving of labs and Corona. I would like to thank you for co-organizing the lab uitje, for supervising students and helping with courses. I hope that you will find or have already found a suitable and interesting job in China. I would also like to thank Professor Bulgaresi and Professor Scheffers for being present at your defense and all the other members of the committee for the time they have put in reading your thesis and being present at the defense ceremony. Regard the obtained dignity as an honor and a privilege, and never forget the attendant responsibilities which you now bear towards science and society.
Sorry, Mr. Rector, you're muted.
Candidate, it is my very pleasant duty, most learned scholar, to congratulate you also on behalf of the doctorate board of the University of Amsterdam with your newly acquired honor. And with that, I declare this ceremony for closed. You're all free to congratulate Yinglan.
Well, congratulations indeed, Yinglan.
Thank you.
Congratulations.
Congratulations and success with your next step or your life now in China.
Congratulations, Yinglan, and all the best for the future.
Indeed.
In for a dinner. We will go to the polder and celebrate it.
That's good.
Unfortunately, Yinglan will not able to join because she's in China, but we will send you images and thank you very, very much for the invitation to go to the polder.
Of course, definitely.
Yeah, many congratulations, Yinglan. That's a really impressive work. I really enjoyed reading the thesis, and I'm always there. Sylvia and Tanneke notice. These are very interesting bacteria, and it's always fun to read about them.
Yeah, definitely. It's not the typical model organism that you normally have.
I'm very happy you like it. Thank you.
I must say, I mean, doing plant development, this is an issue to divide in this plane or the other. So I was reading it with, and that makes all the difference whether you grow in one direction or not, and how cells differentiate. So I never realized actually that bacteria were also, let's say, dealing with this difficulty to divide like this or like that. So you opened my eyes.
Good.
Thank you.
Thank you.
Just a query. What are you doing at the moment? What's your current activities?
Well, I get offer. I start to my new job.
Yeah.
I get a position in a research institute. It's a NGO, and I will do some studies in microbacterium to investigate new antibiotics or some small molecules against the microbacteria.
Mycobacteria. Well, also difficult organisms. But yeah. Very nice. Very relevant. Yeah.
Meanwhile, I was toasting on you.
Yes. Dear Dr. Wang, before I close the meeting, I think everybody had the opportunity to congratulate you. I also want to congratulate you on obtaining your doctor's degree. And I will send you a PhD statement tomorrow, and we'll be in touch about the rest of your diploma and papers. For now, I hope you enjoy this day with family and friends. And I would like to thank everybody for being present and ask the host to close the meeting.
Thank you very much.
Bye.
Bye, everybody.
Good luck.